I'm Richard Axel. I'm a professor of molecular biology and biophysics at Columbia University, and I have the distinct pleasure and honor of introducing today Professor Pierre Chambon. Pierre is a professor at the Collège de France and the founder of the Institute of Genetics and Molecular and Cellular Biology at Strasbourg, and the well-deserved co-recipient of the Lasker Award. Pierre, it is indeed a great pleasure to have the opportunity to talk with you on so pleasant an occasion.
Thank you.
Let's begin with what I perceive as the inordinate and rapid progress that has been made in molecular biology and genetics over the past 40 years. And I bring this up particularly in conversation with you because you, for the past 40 years, have been at the forefront of what I consider to be so many of the major contributions to the field. This begins with the identification of the polymerases and ends with the most recent studies on hormone receptors. Could you briefly outline your career, what led you to these multiple advances, and talk a bit about why the progress has been so profound?
Well, my background is medical studies. I am an MD. I became an MD in '58, and I had started working in the lab a few years earlier because I realized that I will not be a good doctor. And I was interested in biology. I was interested in research. We may come back to that later on. Why are we interested in research and in science? And this was the time, the late 50s, as you know, all of you know, you all know, that the structure of DNA was elucidated in '53, that in the late 50s, studies on the control of gene expression, crucial experiments were done by both in Paris at the Pasteur Institute and in Cambridge in Great Britain, in UK. We knew that this control of gene expression, of course, was fundamental to understand how an organism is formed, how it develops, how it survives after birth, and how at the end it dies. We knew that, and from the very beginning I thought that this was, of course, a central problem, and I decided that I would like to work on that problem for my whole life in some way. And the reason why I start working on RNA polymerase, as you said, is because these are the enzymes which catalyze the first step of gene expression, which is the transcription from DNA into RNA. Nothing was known at that time. RNA polymerase were discovered, in fact, first in rat liver, and then in bacteria, and the initial work was done in bacteria because bacteria is a much better material for purifying proteins which are present in tiny amounts and to characterize these proteins. And I started working on polymerase at the very late 50s and beginning of 80s. And this culminated, well, I did a few things in between, but this culminated at the end of the 60s by the identification of multiple polymerase in animal cells, whereas in bacteria there was only one polymerase which had been identified. And this, I mean, was satisfactory in some way, because going from bacteria to us, to very sophisticated animals, it was clear that there was a problem, and the problem is a problem of complexity. And the question was how, during evolution, this complexity has evolved and made us possible. And the finding of multiple polymerase was one of the first steps in this series of combinatorial events which allowed the building up of the complexity. Having the enzyme, having purified the enzyme, having characterized the enzyme as a biochemist, of course, was telling us very little about their function, in addition to the fact that they had the possibility to transcribe DNA. And we were in some way stuck at the beginning of the 70s. We were stuck because we had the enzyme. We did not have, we knew that the template for this enzyme was DNA, most likely not naked DNA, because the DNA is associated with proteins in nuclei, in what we call eukaryotic cells, which are cells which could be unicellular, but also the metazoan cells which contain a nucleus. So, nevertheless, when you are stuck, you try to get out of the hole, of course. And one way of doing it was to, try to prepare a template, nevertheless, and working with a template as simple as possible, working with viruses, and also to see what could be the natural template, which is present in the nuclei. And so we did some work on that and found that, of course, one could transcribe, with this purified polymerase, could transcribe the DNA from virus, but we did not know what it was meaning, in fact, because we could not characterize from where in the viral genome this RNA was coming, or only with great difficulty, just because we did not have a tool to do it at that time. For chromatin, which is the association of DNA with proteins, then we did a little bit better, and with other people like Roger Kornberg, we could identify the substructure for chromatin, and which is now known as the nucleosome. But still, we could not really tackle the problem, which was how, in fact, at the molecular level, the gene expression, the specificity of gene expression, the specificity which is really striking, because it is the specificity which allow from a single fertilized egg to move to an animal, having the same genetic information in all cells, and still these cells being different, the cell types being different one from the other, some of them being cells for, let's say, the epidermis of the skin, others neurons, others cells from the liver. And this was really where was the problem. And so this was 72. So we had moved from, let's say, the 50s to 72. We have learned few things. We have learned, but the main thing which had been learned on basic problems. What had been solved was how proteins are synthesized. What has been solved, and this was done mostly with bacteria, but also then reproduced for animal cells. But in terms of control of gene expression, we had, with a simple system of bacteriophage and bacteria, we had some ideas about how it could work, but these ideas were mostly mechanical. Okay? We are thinking about interaction between proteins which were either allowing or which were blocking transcription, but exactly how these proteins were recognizing the DNA, we did not know because the structure of the DNA itself, except the double helix, was known, but the actual structure at well, what we call the base, at the base level, we could not reach. So what happened at that time, it was a miracle. At that time, what happened is that within 3 to 4 years, all of the tools, all of the toolkit which was required to go further and to widely open the research which led to this explosion which has been called the genetic revolution. But in fact, the genetic revolution is first a technical revolution, more than a conceptual revolution. I think the conceptual revolution came in some way from the technical revolution. It's because we found things which were totally unexpected that we had to revise a number of, I would not say the dogma, but what we thought, what we were thinking was mainly derived from what we had learned from bacteria. And of course, when you think about bacteria and you think about us, clearly we are different from bacteria. And there was a famous aphorism of Jacques Monod who was saying, what is true for E. coli, for Escherichia coli, is true for an elephant, which is, roughly speaking, correct, but we thought we are never thinking that what is true for an elephant is not necessarily true for E. coli. And this was really the conceptual revolution. So I think it is because at the same time, new technologies like the possibility of cutting the genome, large genome, in pieces and reproducibly, the possibility to take one of these pieces, one containing a gene, to amplify it, to sequence it, to know exactly at the chemical level what is the structure, the possibility to reverse transcribe, thanks to the discovery of reverse transcription, which had been done a few years earlier, to reverse transcribe the messenger RNA. All of these and many others technical advance which really open this very large field and led to this revolution. And we went from which allowed moving from knowing nothing or almost nothing in the 50s to feeling that we were knowing a lot by the end of the 70s.
So let me turn to the remarks that you had mentioned of our late colleague Jacques Monod, who really felt, who argued that an elephant was merely E. coli writ large. Are there principles that have emerged from the understanding of eukaryotic biology which tell us something of the complexity of the eukaryotic organism that have emerged that really are different from the workings of a simple bacterium?
Sure. I think that there was a paradox, and there were several paradoxes, in fact. But one of them was the relatively huge size of the genomes of evolved organisms like mammals or higher eukaryotes, with genomes which were 1,000 larger than the genomes of a bacteria. And nobody knew how to explain that, in fact. It was called the C paradox, and it was paradoxical indeed. One of the ideas was that there was a lot of junk which had accumulated in the genome during evolution, consecutive to a number of events like duplication, which were in some way abortive, and the DNA, since it was not hurting, was just remaining there. nobody had thought that, in fact, the genes could be much larger than required for the synthesis, but apparently required for the synthesis of proteins. In other words, that up to 90% of the DNA of a gene, or what we are calling a gene in animal cells, could in fact not code for the building block of the proteins, not code for amino acids. And one extraordinary, unexpected discovery was the discovery that in fact the genes of animal cells and some of the virus are split, as said, or are mosaic. So they are made up of an alternance of region which code for amino acids and therefore for the protein, and region which are in between and with role which was unknown. Some of it has been found in between, but mostly the role of this region, which are called introns, is still unknown. Functionally, we still don't know whether all introns have a function or not, or whether they are again remnants of evolution. I think most likely introns are also like the pseudogene or some other genetic material, remnant of evolution, and they may be just the witnesses of duplications and rearrangement and swapping of DNA sequence across the genome.
This important observation in eukaryotes that genes are split by large blocks of non-coding DNA is, of course, very profound in considering how genes are built, and offers, as you point out, the potential that the information in between is playing some role. In fact, in a conversation with Francis Crick shortly before he died, Francis was quick to point out that this DNA was coined junk DNA by him, in fact. And junk DNA is quite distinct. Junk is quite distinct from garbage. Garbage is thrown out. Junk is material that is kept, that can be perhaps used later, in this instance, in evolutionary time.
Yeah, I think it's, in some way, I think junk is, well, I think you make correctly the distinction between junk and garbage, because you may keep the junk. In fact, you may keep the garbage also if you don't have a device to remove it. So it's I think if I would have to define it, I rather would take the word of François Jacob, and I would call it tinkering DNA, because it is the DNA on which evolution can really tinker. And one thing which this structure allowed, in fact, with existence of, was to increase the amount of information for making proteins from a given region of the DNA by using what is called alternative splicing. In other words, when within a given segment of the DNA you have a number of sequence segments which can code for amino acid, for polypeptide, you can, and evolution did it, you can in fact splice these piece of information in various ways. And this, the net result is that you increase the amount of information that you can make from a given region of the genome without really increasing the number of genes, because you are, evolutionarily speaking, what happens is that from a given number of genes, through the alternative splicing, you just generate additional proteins. And this together with gene duplication are certainly the two forces which during evolution have resulted in an increase in the number of proteins. You know, when we say that, after all, we are not too different from, let's say, a worm, or from not too different from a fly like Drosophila, because after all, the number of genes is not so different. True, the number of genes, and in fact, I would question what is a gene. I think, in my opinion, I don't know exactly what is a gene now, okay? I mean, if you want to give a definition of a gene, you may say, well, a gene is a region where you have transcription. Okay? You start from a point, you end up to another point, and even sometimes they are not well defined. And this segment of the chromosome which is transcribed, and then there is no transcription on the left, no transcription on the right, this is a gene. Okay? But when you have said that, you don't know anything about the function. And I mean, one has to remember that a gene was initially defined as a unit of function. For bacteria, from neurospora, for other things, it was defined as a unit of function, something which encodes a protein, a polypeptide, an enzyme, for instance. So it was one gene, one enzyme. Now it's one gene, who knows how many proteins. I think there are examples, I don't have them in my head right now, where you can get 30,000 different proteins from a given, I think it's in the nervous system. So that's something that you know better than me.
DS-CAM
right.
gene in Drosophila.
Okay. So this says that, in fact, this process of splicing is extremely important, has been extremely important for evolution, because it enabled the living organisms to work with a much larger information.
Yeah, I think you emphasize a rather important point, and that is there's been considerable discussion about the complexity of the human genome, and surprised that one could identify what are thought to be about 30,000 genes or genetic units. But in fact, the point you make should be emphasized, and that is that genetic complexity is defined not by the number of genetic units, but rather the number of different combinations of units that are expressed, and how those units may vary both in terms of gene splicing and protein modification. Now, the complexity of the genome and the potential number of protein products taken together with the rich but well-defined regulatory mechanisms have led to the suggestion that even in complex organisms, the developmental process is innate, that it's determined. And not only is the developmental process determined, but some have argued that an organism's capacity as an individual and its behavior may be innately determined. What is your thought?
Well, I think for me it's relatively clear. The gene exists, the genetic information exists, but the genetic information itself does not determine an organism. What determines an organism is the interaction between this genetic information and the environment. And this, because around this genetic information, what is built is what is called the epigenetic information. And the epigenetic information, to a large extent, depends on interaction with the environment. It is the environment which will, for instance, decide whether a protein is or is not phosphorylated, which will have a profound effect on its activity. And this is not written in the DNA. Okay. Whether the triggering event which will result in this phosphorylation will happen or not depends on what happens in the environment. This environment is both the cellular environment, which cells are around a given cell, but also it's the outside world. So, we are genetically defined to the extent that without the genetic information we don't exist. But at the same time, we depend on what happens outside of our genome. Which is extremely important, in fact. If you think about what this really means, it means that for we human beings, what we are is certainly determined to an extent that, I don't know, I can't say 50%, but I don't know if it's 50, 40, or whether it's 30 or 60, I have no idea, but at least part of it, a good part of it, and this is obvious to all of us. Okay? You just have to look at your father, your mother, and you resemble your father and mother, and your sister and brothers and so on. So we are determined to some extent by our genes, by our chromosome that we have inherited. But for the rest, we are determined by the environment. And the environment is something which is not genetically controlled. And therefore, if we are gifted for something, there is probably a genetic part in it, and it's well known, the Mozarts in the family were all gifted for music. But I'm sure that there is in the world at the present time, probably there is some Mozart somewhere who will remain unknown because not in the proper environment. So I think it means, and this is something that personally I've learned. In fact, I think I always said that I learned a lot from not only from my own discoveries, but also from the whole evolution of biology. It says that we should not be too proud of what we are as individuals, because the influence of our own will on what we are is probably quite reduced, or a minor part of what we are. There is one thing, if you go to philosophy, Sartre said that l'existence précède l'essence. I think he's right. Well, it means that
existence precedes essence.
Yeah.
The understanding that we have of the full complement of our genes and the way in which we can, in fact, in many instances, in part as a consequence of your work, predict how those genes are going to be regulated in response to the environment has created both ethical and social and at times, unfortunately, political issues that surround the new genetics. And this perhaps began with the overriding concern of the potential for danger associated with recombinant DNA, and then was fueled by the ability to introduce genes into living organisms, and as a consequence, the genetic engineering of plants, and most recently, the issue of stem cells and cloning. what do you believe the role of a molecular biologist is in these ethical and social issues? And we can come to the role of politics in these issues later.
I think our role is to inform the lay people of what we have found and what could be done with what we have found, and to refrain from giving our own opinion on what should be done. Because I think, I'm convinced that my own view of what use should be done with science is not better than the view of anyone in the street.
Do you really believe that? You wish to
Yes,
leave
I
it in
do
the
believe
hands
it.
of politicians and theologians?
You know, there is no alternative. Either you take the power or you leave the power to the others. Okay? But what we have absolutely to do is to inform. This is important. And to inform honestly. Okay? The problem, what has happened is that the scientists they are human beings. And therefore, when they present things, they present their discovery, often they make prediction from what could be done with what they have discovered. Cloning is a typical example. Okay? Whether it will be possible one day or another to clone a human being, I don't know. I think it's not for tomorrow. But we have lived during the last, I don't know now, what, 10 years, 15 years, with the idea that human cloning will be tomorrow, will be an immediate, present risk. It is not. So I think we have to be careful. I think scientists have to be careful. The other thing which also they said, we scientists said when the genetic revolution happened, tomorrow we'll have new drugs for this and this and this and this, so a lot of optimistic predictions were made, forgetting the complexity of the problem which had to be solved before it will be possible. And this explains also to some extent probably why the public is rather deceived of what